Could I get the clicker?
Sorry.
All right, thanks for the clicker. All right, good afternoon, guys. My name's Sujay, as you probably heard. And unlike most high school kids, I work in a lab at the University of Calgary at the Southern Alberta Cancer Research Institute working with brain tumors. So that's what I'm going to talk to you about today, cancer. And the term cancer has a lot of taboo associated with it. It's a term that's used to describe pain and suffering, and it's certainly something that no one wants to hear, especially during a diagnosis. So in terms of cancer, brain tumors especially have been receiving a lot of attention as of late. And a big problem that we're facing in the field of brain tumor research is the fact that we're pumping a lot of money into the field and we're not really seeing much in terms of patient survival. So looking at the screen, there's a number on there, $1.5 billion. That's a 1, that's a 5 with 8 zeros afterwards. And that's a lot of money. And some of you guys might be trying to think of, what you would do if you had that kind of money in your pocket. I'd probably use it towards university education, but I mean, in regards to the number that's on the screen, it's actually the amount of money that's being pumped into cancer research in the States. So this is total funding on an average of over 10 years that the National Cancer Institute pumps into brain tumor research in the United States of America. So this graph shows that. It's on an average of about $150 million a year, which is a lot of money. And that's, again, only counting the US. So that's not including any other countries that fund cancer research, and it's not including third-party fundings from non-for-profit organizations. So in terms of money, the reason we're putting so much money into this is so we can ultimately see something come out on the other end from bench to bedside. We can see improvements in patient survival. So looking at the situation today, we provide money, and money makes our world run. It's what gets everyone going, and it's really necessary to accomplish anything in this world today. So the money going in funds research, and that funds people like me, that funds people, thousands of people around the world who spend hours and hours on end peering over microscopes and working with cell lines. It's a lot of fun. And basically with this research, ultimately what we're hoping to see is we're ultimately trying to translate this into lives saved. But looking at glioblastoma, now this is a type of brain tumor that we work with a lot. It's actually the most common type of brain cancer found in adults. With glioblastoma, a diagnosis of it is really bleak. Now, the statistic on the screen shows that around 6 in 100 people, or in this case 6%, are alive after 5 years. So that's long-term survival. Now, just doing some simple subtraction, that means that 94% of people who get this diagnosis of glioblastoma will die within 5 years, which is a really drastic number, especially considering a cancer that isn't rare, isn't minor. I mean, it's the most common brain cancer found in adults. So we have to ask ourselves, we've spent decades putting money into research. We've spent decades doing research, and we've had numerous advancements in neurosurgery techniques and chemotherapy drugs, and at the end of the day, we're really not in a better place than we were 50 years ago. So we have to ask ourselves what the problem is. So looking at this diagram again, clearly the money's coming in. I mean, we're putting money into this, but we're not seeing something come out. So there's obviously some problem either with the research that's taking place or translating that into actual patient care. So whenever an individual tries to address a systemic problem, I guess the smartest thing to do is to first try to understand where that problem comes from and to try to figure out what the current situation is. So in regards to this, if an individual were to be given a diagnosis of glioblastoma, the most common thing that a neurosurgeon would do is they would go in and try to operate on the tumor. They try to cut out as much of it as possible, assuming, of course, the tumor is operable. And in a lot of cases, it isn't the case, and patients are usually off to a much worse fate. So following surgical resection, as they like to call it, they follow it with aggressive chemotherapy. So these are cocktails and vast concoctions of drugs that ultimately leave a very drastic physiological, physical, and even emotional impact on patients when they go through aggressive chemo and radiotherapy. So at the end of the day, even when we have surgical resection, and again, as I said, even with the numerous advancements in neurosurgery techniques, the therapy is palliative. And what that means is that it's not going for a cure. It's rather a temporary prolonging of life or a reducing of suffering for a short term, and people eventually know that they are going to die at a certain date. So we really have to look at why this is the case. So what I do in the lab is I look at populations of cells called brain tumor stem cells. Now, this seems like an interesting word because you've probably heard of brain tumors and you've probably heard of stem cells, and usually they're heard in different constructs. I mean, I'm talking about brain tumors and I'm talking about how much they suck. But in terms of stem cells, you've probably heard a lot about the good things that people say that stem cells can one day accomplish in the field of medicine. And really what brain tumor stem cells are is that they're a small subpopulation of cells found within a tumor that have added characteristics that are similar to that of stem cells. So what this means is that these cells actually have the ability to form new tumors. They have the ability to migrate to new sites, to metastasize, and to ultimately ensure that the aggressive characteristics of these tumors are met. So studies have shown that these cells are the ones that are responsible for drug resistance, for these aggressive characteristics. And the problem that we're facing with today's therapy is that there's nothing on the market or nothing that's being used in the clinic that specifically targets these cells. So looking at a little bit of a model, now this is credits to my, this is credits to my really terrible drawing skills. We have a glioblastoma tumor. So it's comprised of many, many different subtypes of cells. And within those, one of them will be a glioblastoma stem cell. And if we do conventional therapy, we cut out a chunk of the tumor, we give them a high dose of drugs. If some populations of cells remain, after a certain latency period, these cells can come back and give rise to new tumors that are drug resistant. So clearly these are a problem. So what I do is spend hours and hours on end peering through microscopes. It's exciting. So if you were to take a look through this microscope, you'd probably see something like this. You'd see these little clumps of spheres growing around, and these are what we call neurospheres. They're essentially a model that we use to look at brain tumor stem cells. And basically, each one of these balls will be a cluster of hundreds of, thousands of cells, where a subpopulation of them will be these brain tumor stem cells. And basically, what I do in the lab is I try to take an image like this, where we can see a couple of these stem cells growing, and try and turn it into something more like this. And some of you may not notice anything different, but the keen ones amongst you would notice that there's more little fluffy balls floating around. And you might be asking why that's necessary. And really what I do is, my job is to increase their survival. I'm trying to make them more aggressive. I'm trying to take these cells, and even though they suck as it is, I'm trying to make them suck even more. And that seems really redundant in a way, but the ultimate purpose of that is to A, figure out what keeps these cells growing and what biologically makes them so aggressive. And in regards to this specific project, I looked at a specific protein that's involved in cellular signaling, and I looked at what role that had in maintaining the aggressive characteristics that these cells had. And essentially, I found easy ways to grow these cells, and I found ways to make them more aggressive. But ultimately, where that helps us is when we take away these proteins, or we actually give them inhibitors. And what that ultimately does is that if we can find better ways to target the true chemical factors, or rather the true biochemical factors that make them aggressive, we can actually pave the path towards better, more effective therapy. So the graph on the screen is filled with a bunch of useless scientific jargon most of you guys will probably not end up caring about. But basically what you can see is that as the bars move from the left to the right, they get shorter. And what that shows is that as I'm giving them this inhibitor of a certain protein, their aggressive characteristics are reducing over a concentration-based curve. So one thing to remember is that there are hundreds of projects like this going around in thousands of labs across the world, and there is no such thing as a magic bullet. The work that I've done isn't a magic bullet, and there is no cure for cancer as of yet. And I'm not sure if there ever will be. And the reason I say that is because of looking at this image. And we get this image when we take a patient's tumor, we cut it out, and then we culture them in these flasks. And what we do with the cells then is we put them on these cover slips and give them the ability to just grow on their own, to differentiate. And we get some really, really interesting and beautiful cellular architecture that they create. And after they've done growing, we give them antibodies to stain them using fluorescent labels so that we can better image what types of cells are there based on certain protein markers. And what you can see is each color represents a different protein that's being expressed, and ultimately a different type of cell line coming from the original patient's tumor. So the blue represents cellular nuclei, and the green and the red both represent different types of cell types found within the tumor. So the reason why treating cancer is becoming so difficult is because even within a single patient and a scientist is to go through and better understand how these cells behave on a biological basis, and then get a good understanding of what keeps them going, and then tailor specific medicine suited towards the patient. So I've talked a lot about cancer research, and that's what really sparks my interest. And it might spark your interest as well. And I'd really encourage you guys to get involved with whatever it is. But the problem that we face in today's society is the fact that there are so many systemic issues going around in our globe. And we're trying to apply solutions based on pre-existing concepts. And clearly these concepts aren't working in regards to chemotherapy with brain tumors. And the only way that we can sort of address these systemic issues is to apply a creative and original solution in ways that we might not normally consider. And I think that the people to do that are the people that are in this room, people of our age, people of our generation. I think that young people are the most qualified to solve the most systemic issues. And when I say qualified, I don't mean having a PhD or a master's degree. I mean having a little bit of teenage optimism. And your parents might call it arrogance or ignorance in some cases, but I call it teenage optimism. And I think that together, if we each found our own issue and found our own creative way to find a solution to it, I think that together we'll change the world. Thank you.